All right, we're on chemistry, and I know how much you guys love chemistry. So just realize you're going to have to do a bit of memorization here, and you're going to have to memorize some names and what their names stand for as far as the physics of gases and the chemistry of gases. Dalton is the one that describes how we measure gases or determine their concentrations, and that's that partial pressure thing. So partial pressure came from Dalton. And atmosphere pressure is 760 millimeters of mercury, and the most abundant gas in our environment is nitrogen. But we don't use nitrogen, so it doesn't matter that it's in our environment. Just like X-rays. Can we see X-rays? No, we don't have photoreceptors for X-rays, and it doesn't matter as far as we're concerned. So nitrogen's in our environment in a very high concentration, but we don't utilize it. So it doesn't matter for us. What we not like is this guy here. And carbon dioxide are real important. Water also is important, but that's to remind us that water is inside of our lungs as well as outside of our lungs. So that's Dalton. Henry then came up with a law. He made an observation that if you had a concentration of gases and you had this fluid in a container and you measured the gas concentrations inside the fluid, that whatever what was in highest concentration outside the fluid actually ended up being in the highest concentration of those gas mixtures inside the fluid. And so that's where that comes from. Also, it's important that we actually talk about this because in order for the gases to get into our cells, they always have to travel through a fluid medium. Because remember, we have water lining our little air sacs. So a gas has to go through the water to get through the cell, to get into the bloodstream, which is full of fluid, plasma, before it can get into the cell where hemoglobin is designed to carry it in the red blood cell. So that's another reason why we have to actually know about Henry's law. So he's all about the equilibrium of gases in contact with a fluid medium. So everything always has to go through fluid in order to get into our cells, which are also full of fluid. Cytoplasm is a fluid mixture. We have two areas of gas exchange, and now we're on that topic of respiration. So remember, respiration is gas exchange. Breathing and ventilation is the movement of gases through areas. Now, gas exchange or respiration is going to occur in two areas. We have it external and internal. External is at the area of our lungs, and internal is at the area of the cells. So, blood entering our alveolar capillaries. So this is blood from a pulmonary artery. Is this used or fresh blood that is entering into the alveolar capillaries? Used. Okay. And so our numbers reflect that. Our O2 level is low, it's at 40 millimeters of mercury, and our PCO2 level is high at 45 millimeters of mercury. So that's basically telling you we've sucked as much oxygen out of the blood stream that we can, and we've put our maximum amount of carbon dioxide into it. Now, blood leaving the alveolar capillaries now is considered fresh, and even though it's in a vein, this is the pulmonary circuit, and our PO2 level is 104 millimeters of mercury, and our PCO2 is 40 millimeters of mercury. So we end up with a concentration of oxygen and carbon dioxide in our arterial blood the same as that which is in the air around us. And that's what the cartoon is meant to actually show you too. And I want you to also understand that as we go in, where our used blood is coming in, gases are going to move from an area of high gas concentration to an area of low gas concentration. So we follow that simple diffusion model still with this process. And you may ask yourself, well, if the PO2 going into the capillary and going to exchange with the alveolus is at 40, and the PO2 in the alveolus is at 104, doesn't the PO2 in the alveolus go down as we do this exchange? And I want you to think back or look back at this picture up here. Let's go way back there. This picture here. Take a look at the little capillary lines and then take a look at the size of the alveolus. The alveolus is huge when you compare it to the little lacy capillaries that are covering it, correct? So I want you to realize this is like a huge vat of gas or whatever, fluid or air, whatever works for your conceptualization. This area has a whole bunch of oxygen in there. And this capillary that has just a small amount of oxygen in there, where we're going to actually saturate it on up to 104, it doesn't matter that we're taking all this oxygen out of here because it's so huge. You guys follow me with this? If I'm going to take a few pennies from a millionaire, does it matter? And so that's what's happening. The other thing that's also important, the alveolus is in contact with our air, right? That's the air that's around us. As long as we don't block our airway, we're always purging it. Isn't that what ventilation is all about? Always kind of refilling it, making sure it never gets too low. And so you have to realize that as we travel along and become 104 and a PCO2 of 40, we're not greatly impacting this giant lake of gases with this exchange. And so that's why when we come out, we will actually have the same PO2 and PCO2 that the alveolus has. So that's the story of alveolar gas exchange. And it occurs across the respiratory membrane. So remember, simple squamous epithelium, basement membrane, and simple squamous epithelium, which is also endothelium. Now, internal respiration is occurring at the level of the tissues. So it's at the cellular level. And so it's basically the gases are moving in the opposite direction now as they were with external respiration. So we needed O2 at external respiration, so it went in, and we needed to get rid of carbon dioxide, so it went out. Now we're at the cellular level, and we have all of this O2, and the cells don't. And the cells have all this CO2, because who produces CO2? Cells. Where is CO2 always going to be the highest then? In the cells, right? The ones that are always producing CO2 will always have the highest CO2 level. And so the CO2 that you find in the interstitial fluid is just a reflection of the CO2 that's being produced by the cells. You remember in capillary dynamics, we talked about how we put all this fresh interstitial fluid around our cells, and then we took out used interstitial fluid on the other end of the capillary bed. So you have to realize there's always this flushing that's going on, but the cells that the producer of carbon dioxide will always be the one that always has the highest CO2 level, correct? Yes?
So when the blood leaves the lungs, it's at 104 milliliters of mercury?
Correct.
So
it's partial pressure.
95.
Pardon? Okay. So here it is. If it's at 95 in that area, I didn't even notice that, so thank you for pointing that out. If it's at 95 in that area, it means that we've actually had some gas exchange somewhere along this capillary, correct? So, but going into the capillary, you're right. For us, when we first go in, it should be noted as 104. But this is actually showing like something has happened and we've extracted some from that capillary. But as we go along, we'll go from 104 to 90 to 80 to 60 to 50 to 40, because remember that's kind of the lowest PO2 that we end up showing that we've had maximal extraction of oxygen from the bloodstream. So as we move along, we will drop our PO2 down to 40 at the venule end of the capillary bed, and our PCO2 will be up to 45. So it's really interesting. There's a wide range here for our oxygen, PO2 of 104 down to 40. But for our CO2, it's 45 to 40. And the range is much smaller. And what's fascinating is that it's the CO2 that's the most important thing for us in our respiratory system. CO2 concentration is a very big driving factor. And that's because CO2 concentration will actually determine whether or not you have acidosis or not. And acidosis is a bad thing as far as your central nervous system actually is concerned. So we need our PO2 to go into the cells, and we need our CO2 to go out of the cells into the bloodstream. So that's internal respiration. Let's take a look at how the gases are actually carried around in our bloodstream to get to the cells. So we're interested in O2 transport and CO2 transport. What's interesting is, as far as the two gases go, O2 is less soluble than CO2 in liquid. I know that goes against what I told you earlier about Dalton and Henry, but O2 is less soluble than CO2. And because of that, and we need O2, we have acquired a different way to carry oxygen to our cells so that we can give them the oxygen that they need. And that's what hemoglobin is all about. Hemoglobin is inside our red blood cells, and our red blood cells are bags of hemoglobin that are designed to carry oxygen for us. And we know per hemoglobin molecule that we can actually carry up to 4 oxygen molecules, O2s, which is an oxygen molecule per hemoglobin, because we have the 4 heme sites. So if you are in an area where the oxygen level is low and you have hemoglobin present, the hemoglobin will not bind as many oxygens because the oxygen concentration is not so high. But if you have a high oxygen concentration, the hemoglobin molecules will bind more and therefore carry more oxygen. And that's why we can get our PO2s down to 40, because we're in areas where O2 levels are low, and so hemoglobin's not going to actually hold on to that oxygen in low O2 level situations. And that's actually important, because we don't want hemoglobin carrying a bunch of oxygen when O2 concentration is low, do we? We actually want hemoglobin to actually give up as much oxygen as possible in areas where O2 concentration is low. You guys did some fills, exercises about pH and things like that with hemoglobin, and so let's go ahead and take a look at some of these notes here. Higher temperatures. I want to ask you a question about this. What do you think the temperature is like inside the alveolus compared to the temperature that's in our muscular tissue? What do you think the temperature is in comparison? Which has a warmer temperature, in other words? Inside the tissues, inside the muscle tissue, for example. Because what's the lungs in contact with? The inside of the lungs. It's in contact with the air. So, which makes good sense? Do we want hemoglobin to bind more at a higher temperature or for oxygen, or at a lower temperature for oxygen? Which one? Which temperature do we want for hemoglobin to actually hang on to its oxygen molecules? Lower temperature, which is perfect, because it's cooler in the airways than it is in the tissues. And so higher temperature means more O2 delivery to warmer tissues. And if the tissues are warmer, aren't they more metabolically active? And would that make sense if you're more metabolically active, that you need more oxygen? And so at the area of the lungs are cooler. So hemoglobin binds more O2, which is perfect. That's really great. So I'm going to talk about binding of oxygen and dissociation. So dissociation means we give it up. And affinity means that we like and therefore hang on to it also. So I'll be using some of those terms. higher acidity, which means low pH, so a high acid environment. Let me ask you, what do you think the acidity level is in metabolically active tissue versus tissue that's not doing too much, so therefore not so metabolically active? What do you think the pH is like between the two? Yeah, high activity, lower pH. When you exercise excessively, what do you produce a lot of? Lactic acid. And so that's what's really good too, because now we actually, when we have a low pH, which is a high acidity, we have, again, more O2 delivery to low pH. And in order to do this, hemoglobin and O2 has to disassociate. It has to give it up. So if we are more alkaline, then hemoglobin will actually hang on to or bind more oxygen. And another chemist or physicist you have to learn the name of is Bohr. Bohr found that if you had a lot of hydrogen ions around, that enhanced oxygen unloading from hemoglobin. And so we also found that hemoglobin, interestingly, functions as a buffer somewhat. Do you know what a buffer is for in a chemistry experiment? It's to minimize pH changes, right? So what's interesting, hemoglobin functions as a buffer as it can bind hydrogen ions or protons. So if you have a whole bunch of protons and hemoglobin's in there, you can kind of take them out of solution and bind them so we aren't as acidic, and that's what's really nice. And that's why when we have hydrogen ions present or protons present, we enhance the unloading of hemoglobin because we end up with high proton loading causes conformational changes in the hemoglobin molecule so that it gives up or unloads O2. So when I say conformational changes, I want you to think of the transformers. You know, they change their shape, and because of the change of the shape, it no longer can actually allow oxygen molecules to actually be bound to it. Carbon monoxide is CO. Carbon monoxide poisoning is a bad thing. That's why we have carbon monoxide detectors in our home, because carbon monoxide is a gas that is colorless and odorless. And so when it's in our environment, we don't know about it. And carbon monoxide, even in relatively low concentrations, is really, really bad for you. And that's why we do have the carbon monoxide monitors in our home, because carbon monoxide binds to the same side as oxygen, preferentially. So what does that mean? If you have hemoglobin and you have having it come into contact with a mixture of carbon monoxide and oxygen, what do you know about that hemoglobin molecule? It's got a bunch of carbon monoxide instead of oxygen. So it has a higher affinity for carbon monoxide. And that's what's important, because the other thing about this high affinity for carbon monoxide is when it binds carbon monoxide, it loves it so much it won't let it go. It's kind of like a person with an obsession over somebody. They just won't let them go. So carbon monoxide and hemoglobin is an obsessive reaction, so to speak. So it binds carbon monoxide and it won't let it go. So that means if you're in a even if you're in a low carbon monoxide environment, as that hemoglobin molecule comes back to the airway and it already has some carbon monoxide on it, it will bind even more carbon monoxide. And that's why carbon monoxide is so bad for you, because your hemoglobin molecule won't let it go. Well, as your hemoglobin molecules continue to fill up with carbon monoxide, what's the problem? You're not carrying the oxygen to the tissues that need it. And so what are you doing to your tissues? You're cutting them off from their oxygen. You're killing them, And so your body will note that, and it will start to make you feel nauseated, sick, lethargic, and all kinds of things to try and get you to go to the doctor or get out of the environment, actually, because that's part of the problem. You need to get out of the environment where the carbon monoxide is. And so it's an interesting phenomenon, because whenever somebody has carbon monoxide poisoning, they are a bright red color. They are not cyanotic. They are not bluish. They are cherry red. So CO So when somebody comes into the emergency room and they're nice and brightly colored, they have a nice flush to their face, and they say, I feel really, really bad. And the triage nurse is looking at them saying, well, you look pretty good to me. I don't know what the big rush is, right? And it has no idea that that person is really, really in dire straits and sick, because they are not getting enough oxygen to their tissues for adequate perfusion and oxygenation at the cellular level. And so when somebody has really bad carbon monoxide poisoning, do you guys know what you do to treat carbon monoxide poisoning? Besides, obviously, you must get carbon monoxide out of the environment, correct? Because if you don't get carbon monoxide out of the environment, we'll just keep loading up preferentially with carbon monoxide instead of oxygen. So carbon monoxide must be removed from the environment. So what else would you do besides that to treat somebody that's really bad with their pure oxygen? Not just a little bit of oxygen, pure oxygen. And one of the things that you don't know yet, and you will later on in your clinical experience, is that pure oxygen is actually toxic to the lungs for prolonged exposure. So we live in a 21% O2 environment, and pure oxygen means 100%. And you can do that for a little bit, but after a while, you actually start burning up your lungs. So it's not really good for you. But when somebody's got bad carbon monoxide poisoning, that pure oxygen is also not going to be all that best at getting the carbon monoxide off of our hemoglobin molecule. And so in order to treat somebody with bad carbon monoxide poisoning, you put them in what I call an oxygen pressure cooker. And you put them in a hyperbaric chamber and give them 100% oxygen in a hyperbaric chamber, and you literally try to push carbon monoxide off of the hemoglobin molecule with hyperbaric oxygen. So it's a insidious pathogen. You don't realize that it's actually in your environment. So that's carbon monoxide story. Now let's talk about the carbon dioxide story. We can actually carry carbon dioxide in our blood 3 different ways. A little bit of it is actually in the plasma. So in other words, it's more soluble in the liquid medium of our blood than oxygen is. We can also carry carbon monoxide bound to hemoglobin molecule. And about 20% of it is bound to hemoglobin. And its binding site, carbon dioxide's binding site, is different than O2's binding sites or carbon monoxide's binding site. So it's a different binding site. And then when we are carrying carbon dioxide on the hemoglobin molecule, it's called carbaminohemoglobin. I have to always stop and think about it as I try to say that. The best way that our body has evolved to carry carbon dioxide in our system is as the bicarbonate ion. So 70% of carbon dioxide is carried as a bicarbonate ion. Let's take a look at this chemical reaction. Carbon dioxide made at the cellular level plus water, which there's plenty of that in us, in the presence of an enzyme called carbonic anhydrase. That's the enzyme that catalyzes this reaction, where CO2 plus H2O will give us our carbonic acid. H2CO3 is carbonic acid. Carbonic acid, anything that's called an acid, means that it separates or dissociates into a proton and the other part. And so carbonic acid will then want to separate or dissociate into a hydrogen ion, which is a proton, and the bicarbonate ion. And so that's how we carry most of our carbon dioxide around inside of us. What's interesting, carbonic anhydrase is in very high concentrations inside our red blood cells. And so this reaction will occur at the cellular level, and it will go into the plasma and into the red blood cell, and then we'll get our acid formed, and we'll get our protons and our bicarbonate ion. Now, the other thing that's important is that when we get to the level of the lungs, this reaction is reversed. And so we'll end up with carbon dioxide to off gas. In other words, to put into the alveoli. So this is really cool. This reaction goes two ways. And carbonic anhydrase is the driving factor for this actual reaction. So this is how we carry the bulk of our carbon dioxide around in our bloodstream in this form of the bicarbonate ion. Another guy. Now we have 4 names. So Haldane was involved in noticing that when we have a low PO2, partial pressure of oxygen, it will increase the amount of carbon dioxide carried in the blood. And this is important because at the tissues, we do have low PO2. So we can actually carry more, because of our low CO2, more CO2 as it is bound to hemoglobin, and also because of the tissue pHs. At the lungs, where we have high partial pressures of oxygen, PO2, can get rid of more CO2 in this area, especially the CO2 that's bound to hemoglobin, which is great because we want to unload our carbon dioxide at the level of the lungs. So the higher the PO2, the less CO2 that the blood carries. The lower the PO2, the more CO2 the blood can carry, which is actually what used or deoxygenated blood is. The higher the PO2, the less carbon dioxide that the blood will actually carry, and the lower the PO2, the more more carbon dioxide blood will carry. And now let's go into just a little bit of discussion of carbon dioxide and blood pH. More CO2 means lower pH. So our cells are always producing carbon dioxide, so the environment around them is always more acidic unless we keep flushing it out. So the more CO2 in any system, the lower the pH. So CO2 and pH are directly related, and they're inversely related. Correct? So it's an inverse relationship to pH. What's pH's relationship to proton concentration? Or hydrogen ions? The lower the pH, the what the hydrogen ions?
Higher.
The higher. So you have to remember there's this little inverse relationship between some of these areas. Okay. HCO3, which is the bicarbonate ion, produced in the above reaction, acts as a buffer when we have a lot of protons present. And that's because when we have HCO3 and protons, when we have this as a high concentration, we can drive our reaction in the opposite direction. Another thing to talk about before I get any further in this is strong acids and weak acids. Do you guys know what the difference between a strong acid and a weak acid is? Correct. A weak acid doesn't want to actually, like this guy here, he is a weak acid, and he doesn't really dissociate very much and produce a lot of protons. A strong acid, like hydrochloric acid, means that it wants to immediately dissociate or separate and produce a bunch of protons, and it's the proton concentration which is what makes you an acid. So a weak acid means that, like vinegar is a weak acid compared to hydrochloric acid, correct? And so what that means is when you check the proton concentration, it's not as high, which means that this reaction isn't driven. Let me erase a few things here. If the proton concentration isn't very high, this reaction isn't very high also. I'll put a line through that, because that's what that's meant to be, as a line through that. That reaction is not very high. So even though I say it's carbonic acid, the only time it's really an acid if it dissociates and separates and produces a bunch of protons, correct? The more it dissociates or separates and produces protons, the stronger the acid is. And that's an important thing. This is a weak acid. So if for some reason we have a bicarbonate ion, such as sodium bicarbonate, baking soda for example, we can use this side here as a buffer for protons and push this reaction in the opposite direction where we get the weak acid. So bicarbonate ions will act as a buffer for a lot of protons. The other interesting thing is this weak acid also acts as a buffer when pH is high. A high pH is an alkaline situation, and in order to neutralize an alkaline situation, we add some protons to it. An acid and a base is a neutralization reaction, correct? An alkaline is a base. And so if we actually have an alkaline pH, we can put a little bit of this weak acid in there, and it will help neutralize it or buffer it. Because we can use this guy and this guy to help buffer things, our blood pH doesn't move much beyond 7.4. Let's do some examples of our weak buffers here and take a look at some strong acids and strong base. Okay, NaOH, sodium hydroxide. That's a very strong base. So which one of these two guys up here do we want to go ahead and mix it with? H2CO3. So when we put a little bit of our weak acid, we actually will then end up with sodium bicarbonate, which is a weak base, plus good old-fashioned HOH, which you guys know is the same thing as H2O. Now let's take a look at HCl, a very strong acid. Which one of those buffers do we want to actually mix this with? The bicarbonate, right? And in our system, what cation is in high concentration outside of our cells? Sodium. So let's go ahead and add a little NaHCO3, because that's one of the ways that bicarbonate ion is actually kind of hanging out with a little bit of sodium. And we'll get NaCl, which is salt, plus H2CO3, which is a weak acid. And this bicarbonate system, where we can use the bicarbonate ion and carbonic acid acting as a buffering for us, is very important in actually keeping our blood pH pretty darn stable. The important thing to always remember, the more CO2 we have in our bloodstream, the lower the pH or the more acidic we are. That's always a thing to keep in mind, because we're going to go into control of ventilation.
For someone that's like really sick for a long time, does that just mean like they're really like an athlete or something, like they are always working out? I didn't hear the first part. If someone has like really acidic blood for a long time, does that just mean like they have less CO2 in their blood?
If somebody has very acidic blood, what do you know about the CO2 concentration? It's got to be high. So if they have an acidic pH in their blood, their CO2 concentration is high. And that's actually very common in people with emphysema or COPD. Their CO2 concentration is high because they don't have proper respiratory membrane for gas exchange. Their O2, does anybody know what the O2 concentration is for somebody with emphysema that's not on supplemental oxygen? Why would they be on supplemental oxygen? Because their O2 concentration is low, and that's because they have actually had destruction of their respiratory membrane. So if their O2 concentration is low, their CO2 concentration is elevated. One of the things that's real important to actually help to mitigate, you guys know mitigate damages means? To minimize damages. Your kidneys will help you pee out the acid. So the kidneys are the other factor that's going to help you out. But if you are acidotic, if your pH is acidotic, we're going to talk about how you should actually correct that in just a moment. Ventilatory centers, let's go into this. It's in the medulla, and that means medulla oblongata, which is at the base of the brainstem, correct? What else is in the medulla oblongata that keeps our basal things going? Heart rate and blood pressure, correct? So also our respiratory center is in the medulla oblongata. And that has neurons in there that fire spontaneously like the nodal cells of our heart. So the medulla oblongata has a pacemaker for your ventilatory rate. So that's an important thing to actually realize. And when they fire, impulses go down these two nerves. One is called the phrenic nerve, and the other are called the intercostal nerves. What do you think the phrenic nerve supplies? Just a guess. What helps us ventilate, people? Diaphragm. Oh, that was rough. So diaphragm. So it goes down the phrenic nerve to stimulate the diaphragm to contract. And then the intercostal nerves, what are they going to actually stimulate? That helps us with ventilation? Our external intercostal nerves. Or muscles, excuse me, intercostal muscles. So they're spontaneously firing, and relaxed ventilatory rate is around 6 to 8 respirations a minute for most people. If you're in a really super relaxed state, you can even get it down to 4 ventilations per minute. Should have said respirations, I should have said ventilations. Okay, the expiratory group, there actually is one, but it is only active during forced expiration. And it goes to our internal intercostals to actually pull our rib cage down to actually force air out. Up from the medulla oblongata, there is the pons. That's the next guy going on up. And it actually does have some instructions that it can do to our ventilatory rate. And so we can say it fine-tunes the instructions from the medullary center. So it actually kind of supplements them. And in the pons, we have an area called the pneumotactic center and another area called the apneustic center. Which one is active when you're sleeping? Apneustic center. And so the apneustic center is what's helping you to have nice, slow, deep breaths when you're sleeping at night. And that's when it's activated. And the pneumotactic center, increasing your ventilatory rate, which is would be very normal for you to do when you are actually awake and working out, doing things. Other things that can control ventilation rate as we're moving up the stack, so to speak, higher brain centers, hypothalamus, and what this is actually referring to, emotions. Can affect your ventilatory rate. So when you're laughing and you're crying and you're sobbing, there's a very different ventilatory rate that's actually occurring with those emotional aspects. Cortically, that means your thinking thing. When you're being cerebral or you're very cortical in your thought processes, what can you cortically do to your ventilatory rate? You can will yourself to breathe at a certain rate, correct? You can hold your breath, you can hyperventilate, you can do all kinds of things. So cortically, you can think it. However, if you want to hold your breath, how long can you cortically, will your cortical centers override the other centers? No, over time, the cortical centers are going to give way. And that's indeed another reason why when people drown, when they're submerged underwater, they're holding their breaths. They want to hold their breaths, but they are unable to completely hold their breaths because the medulla oblongata will tell you, I'm going to make your diaphragm contract. You can hold your breath as long as you want. Close your upper airways, but we are going to contract the diaphragm, and you're going to ultimately go ahead and ventilate, unfortunately. Chemical factors impact on your ventilatory rates. PCO2, remember that's the partial pressure of carbon dioxide. That is our most potent chemical control, big time, because it affects pH. So we have 2 terms associated with our PCO2 levels of our bloodstream: hypercapnia and hypocapnia. Hypercapnia means we have too much CO2. Hypocapnia, hypo or little, we have too little CO2. First off, how can you make hypercapnia? What's a way you can create hypercapnia? Hold your breath, right? When you hold your breath, what happens to the carbon dioxide level inside your bloodstream? It goes up. So you cause by holding breath. So if you hold why would you hold your breath? Yeah, you're swimming or something along that line, yeah. Or it's a game you're playing with somebody, see who can hold their breath the longest kind of a stuff, yeah. So that's one way you can do that. So if your CO2 level is getting too high, what would your body want you to do to fix it? Hyperventilate. So hold your breath. That means there's no gas exchange occurring externally, no external respiration occurring, and so CO2 will build up. And all you have to do is hyperventilate, and you're literally blowing off CO2, correct? Isn't that what hyperventilation does? Because you won't increase your O2 level, but you'll be blowing off CO2. Hypocapnia, too little CO2, so obviously you cause this by hyperventilating. And so, and what, does anybody know what hyperventilating does to how you feel? Yeah, lightheaded, a little queasy and stuff like that. So what this does is this actually causes cerebral vasodilation or constriction. What do you think? Constriction. So the blood vessels to your brain will vasoconstrict right on down. And they'll vasoconstrict long enough to make you faint, pass out. Because when you faint or pass out, whatever you're doing Fulio will stop. Yes? And so it causes causing dizziness and even fainting. If you're hyperventilating bad enough, okay? And so then when you faint, what's really nice about fainting is you fall down and everything's on a nice even plane with the gravity, correct? You're laying down, which means then nobody has to work really hard, right? So you're laying down, nobody has to work very hard. And then, so you watch the person and do they start breathing right away? No, because if you hyperventilated and you blew off all that CO2, what have you done to your blood pH? You've made it go the opposite direction. So you have to wait until enough CO2 accumulates to actually initiate the ventilatory drive for you. So that's what's really interesting about hyperventilation. And kids are interesting little creatures, aren't they? They like to manipulate their parents as best as they can, and they have very few tools to manipulate their parents with. But they sometimes figure out their ventilatory rate can be a form of manipulation. And I'll hold my breath until I pass out, right? And all you have to say is, okay, I'll stand here and I'll catch you, which won't make them happy at all, right? Because they're trying to punish you for something. And then the alternative would be, I'm going to breathe so fast I'm going to pass out, and you can say, okay, I'll catch you, when you fall. And they won't like that either. And I've never known any child to really be very successful at actually causing themselves to hyperventilate so much to where they actually did pass out. But if they do pass out, remember, as a parent, if you do have a very willful child, there are such things out there. If you do have a very willful child that is going to show you, as I said, you just make sure that you're there to catch them on the way down, so they don't hurt themselves. And after they lay down, you have to realize that they won't start breathing right away because they blew off too much CO2, and we have to let some of that CO2 accumulate, and they will start breathing. So you just watch their color, and they'll get back to normal. Those poor little kids. And the last thing that we're going to talk about before we're done with our lecture, and therefore we're out of here, is PO2, partial pressure of oxygen. We actually do have chemo sensors or receptors in the aortic sinus, the aortic bodies, and the carotid, the aortic arch, the aortic arch and the carotid sinus, and they're in the areas that they call the aortic bodies and the carotid bodies. So they measure PO2. And so PO2 can impact breathing or ventilatory rate, but has to be extreme before it actually has an impact. So O2 levels have to fall considerably. One of the things that you have to realize is that if our PO2 on the venule end of a capillary bed to the tissues is at 40, then there was an area that was actually at a PO2 rate of 50 somewhere that was able to extract enough oxygen to survive. And so one of the things that's really important is that you can tolerate bigger fluctuations in O2 than you can CO2, because the CO2 will impact your pH or acid level. And this is well known for any of the people that actually do any sort of competitive diving or prolonged diving, or in the military where they have to dive as maybe a Navy SEAL, because they will take them out there and they will put them out in the water, and they will actually have their they'll lower the individual's PO2 levels to around 60 in the individual before they actually find that they're actually impaired, exhibiting signs of impairment in their ability to function. So PO2 levels can actually, we tolerate wide fluctuations of PO2. And that's also known for, as we were just talking about, with COPDers, because people with COPD or emphysema, they come in and their blood PO2 levels typically in the 70s and 80s. What's most people's PO2 when they do that little pulse oximeter on them? They're in the 90s or 100. So people with emphysema are coming in and the little pulse ox is reading 70s to 80s for their PO2, and they're okay. They're not running marathons though, are they? No, people with COPD are not that very active. And then sometimes they'll be on supplemental oxygen, but not always. Because the other thing too, if the PO2 level is low, what do we already know about the CO2 level, the PCO2 level? It's going to be higher than normal. And you have to realize that these levels got there overnight. No, they got there slowly over time. And that's the beautiful thing about our human bodies. They are constructed to compensate for gradual changes. And so as that individual was gradually losing respiratory membrane or lung function, they were getting lower PO2 levels slowly and higher PCO2 levels slowly. And then the kidney, they actually started to actually breathe faster first, right? As the PCO2 goes up, what do you know the ventilatory rate will want to do? It will want to go up too. So that's important. If your PCO2 goes up, that means you're becoming acidotic, and that means it's going to stimulate your ventilatory centers to cause you to ventilate more. When I say stimulate ventilatory centers, I mean to actually ventilate more. So as the PCO2 went up, initially these people were actually breathing faster, but they could not, no matter what they did, they could only sustain a certain ventilatory rate till they caused themselves to fatigue. And that's the other thing too, is breathing faster for a long period of time is very energy intensive. And so people who actually do get emphysema have a tendency to be a little bit on the thinner side. There's always the blue bloaters of people with chronic bronchitis, but that's a totally different story. But they'll get bigger chest walls and they'll lose weight because of all the energy that they have to expend to increase their ventilatory rate. And so after a while, another system will actually kick in for them, and that's the kidneys. And the kidneys will actually help them to preferentially put acid into their urine, the filtrate, so that they will actually start peeing out acid. And the kidneys will also help to create bicarbonate. And the bicarbonate is the buffer that helps the acidosis. So when somebody's coming in with a low O2 and a high CO2 and they're fine and you put them on oxygen, one of the things you have to be careful with is that you will actually start to slow down their ventilatory drive. And that's one of the reasons too why people with COPD have to be very careful with their oxygen supplementation. The more oxygen you give them, the slower they want to ventilate. And what does that mean to their carbon dioxide levels? They actually start to go up. And so it's a very fine line that you actually have to walk when people have COPD and supplement them with oxygen. So that's all I have about our respiratory system. That is the end of all the material for exam 3. Do you guys have any questions about any of the material before I set you free? Yes.
For the bicarbonate reaction, did you say carbonic anhydrase is an enzyme for both directions?
Yes, carbonic anhydrase is the enzyme that catalyzes the reaction in either direction. If you have too much CO2, carbonic anhydrase will help you move to the acid and then the bicarbonate ion. So concentrations will actually direct which way the reaction goes. Yes.
So you were saying, I'm just going to verify, if you're hyperventilating, you're ventilating more, it's decreasing your CO2?
Correct. If you hyperventilate, you're blowing off more CO2. And if you hypoventilate, you're accumulating CO2. And CO2 is the biggest controller of pH and ventilatory drive via the respiratory system. When somebody has diabetic ketoacidosis, what is their ventilatory rate? High. And the reason for that is why? They're trying to blow off CO2, trying to raise the pH, correct? So they're trying to have a respiratory compensation for a metabolic acidosis. So acidosis and alkalosis can be caused by two different things. You can have it caused metabolically, because isn't that what's happening at the level of the tissues? When carbon dioxide levels are up, the more metabolically active you are, the more carbon dioxide you're going to produce. But that's not a ventilatory thing. That's a metabolic thing. Lactic acid, for example, isn't that a metabolic process? So metabolically, you can have an acidosis, and your body will try to compensate with it via a ventilatory mechanism, correct? And so if somebody is in diabetic ketoacidosis and they have a normal pH, you already can know what their ventilatory rate's like, correct? They're huffing and puffing. But if somebody has an alkalosis, and let's just pretend it's metabolic, it's very unusual to have a metabolic alkalosis, what is their ventilatory rate going to be? Very slow, because they're trying to get that pH all the way back to 7.4. So if they're alkalotic, we want more acid, right? And so the way to make more acid is to hold your breath, increase your carbon dioxide concentration. And when you increase the carbon dioxide concentration, the more you increase it, the more you drive the reaction in that way. Okie doke? That's the end.